You
Yes,
start
sir.
recording. Okay. Now, Aparna, you can start.
Start, sir. Sir, I am audible.
Yes.
Good afternoon, everyone. Welcome to today's session, Empowering Indian Microbiology, Global Influence of IUMS and ICSP. It is my distinct honor to introduce distinguished person Dr. Kamlesh Jangid sir. He is a trained microbiologist and expert in the field of microbial systematic and ecology and soil science. Dr. Kamlesh sir completed his education in microbiology from University Pune in 2005 with the PhD from the National Center for Cell Science (NCCS). His postdoctorate in soil microbial ecology was with Professor William Whiteman at the University of Georgia (UGA), Athens, USA. He returned to NCCS as a scientist in 2011 to build the world's largest microbial culture collection, the National Center for Microbial Research (NCMR). In July 2020, he joined ARI as a full-time scientist in the bioenergy group and as a curate in charge of MACS collection microorganism (MCM). So, please, I will request to continue this session, Dr. Kamlesh sir.
Thank you, Aparna. Thank you, Dr. Deshmukh, for inviting me to present what we are dealing with the IUMS. I'll quickly share my screen. I hope you are able to see the full screen. Okay.
Yes, sir. Yes.
Great. So I will not take much time. I've slightly modified the title of my talk today from Empowering Indian Microbiology to Empowering Microbiology in India, and because of the global influence of IUMS and ICSP. Before we begin the actual proceedings, I would like to take you through the history of why we are here today. So why are we discussing this today? We are discussing this because when I was attending the culture collection meetings in China in the month of November last year, there were a lot of organizations that were represented at the World Data Center for Microbiology microorganisms meeting, and it was quite surprising that none of the Indian microbiology societies was represented at the WDCM. I was part of this meeting simply because I was president of the Burgess Society, so I was attending it on that behalf. And this idea popped up in my mind that why not have a society participate in this. So I contacted Dr. Deshmukh on the 4th of November when this idea conceived. And on the 4th of December, we submitted our application to the IUMS. Dr. Deshmukh provided a lot of documents that we required for getting registered as a society with the IUMS. And then the application was approved by the IUMS pending a final financial committee review. And on 8th of February, we received our first financial dues, and the dues were about $1,400 based on the large size of our membership. But the memberships are typically calculated considering the Western world, and the dollar conversion was not factored in. So we requested, I basically connected with them again, and I requested for them to revise our fees. And finally, on 28th of February, they revised our invoice to $100 annually. And with that, membership fee, we became a member, and the payment was made on the 3rd of March. And gladly on the 11th of March, we received the official certificate of being a member of the IUMS. And the certificate basically said this is to certify that the Microbiologist Society India has been granted full membership of the IUMS. The IUMS is a voice of microorganisms, bringing together regional and national societies of microbiology through the world. And this was very, very unique for us because that was the main reason why we wanted to be a part of the IUMS. Now, to give you a brief about what IUMS is and how many member societies are part of this body, I've only listed from India. IUMS member societies from India, as you can see, there are only two member societies from India. The first one is the International Science Academy, and the second one is the Microbiologist Society in India. Now, this was, very encouraging that from India, there was not much representation of microbiological sciences across the world, whereas if you look at the population our country represents, we represent about 20 to 25% of the population in the world today. And if you look at the number of microbiologists that are in the country, you can just imagine the size from the depth of our WhatsApp groups, our Telegram groups, our LinkedIn following, and so on and so forth. So we can very well say that a major chunk of microbiology was not being represented at the international forum. And because of this, we wanted to be a part of the IUMS. So what is IUMS? IUMS was created as the International Society for Microbiology in 1927 on April 28th at the Pasteur Institute of Paris. And as of 2008, during the formation of the ISM, there were 34 persons from 15 nations which were listed as the foundation members of this society. Now, in 1967, it became the International Association of Microbiological Societies within the International Union of Biological Sciences. And eventually, it became independent as the IUMS, or the International Union of Microbiological Societies, as it is known today. And it became a member of the International Council of Scientific Unions in 1982. And it represents 104 national scientific bodies and 29 international scientific unions under the ICSU. So as of today, there are now 31 international scientific unions which are within the ICSU. Now, IUMS is the global voice of microbiology, and that's primarily because it comprises of about 117 microbiological societies and national committees from 67 countries and one international organization. These are full members, we being part of the union very recently. There are also associate members of the society. There are 7 microbiological societies which are associate members and 11 multinational organizations which are part of this global voice of microbiology. Now, as you can see, the spread on the map, the blue highlights are the countries that are part of the IUMS. So if you leave aside Middle East and parts of Southern America, Africa, most of the other world is part of the IUMS. And because of its broad contact with thousands of microbiologists worldwide, the IUMS can be regarded as the global voice of microbiology. And a major portion from India was lacking for it. So we wanted to be a part and raise our voice on this international forum. The mission of IUMS is to foster and promote international exchange, dialogue and collaboration amongst the international community of microbiologists, which are involved in the discovery and applied sciences, as well as in the development of policy. The major part is they seek to align all microbiological societies in raising awareness of the importance of microorganisms. And we, as the Microbiologist Society India, one of our primary focus is also to raise awareness about the importance of microorganisms, whether be it at the school level, at the college level, at the teaching level, or at the industrial level in research and development. So regardless, because our alignment with the IUMS objectives and mission is very, very clear, it made all the more sense to become part of this body. Some of the objectives of IUMS are to promote the study of microbiological sciences, initiate, facilitate, and coordinate research and other scientific activities. It also ensures the discussion and dissemination of the results of international conferences, symposia, and meetings. And finally, it represents microbiological sciences in the ICSU and maintains contact with other international organizations to see what is happening. And how it achieves these objectives is through conducting these annual IUMS conferences. The most upcoming, the October 2024 IUMS conference, or IUMS 2024, will be held in Florence, Italy from 23rd to 25th October. So this is one of the key factors where our society, as a member of the IUMS, will get a discounted registration for participating in the IUMS. And it is not just about having a discounted registration. It is also about participating in the IUMS larger efforts. And our society, MBSI, has already been doing it for the past many years, these outreach programs and activities, which are also one of the major focus areas of the IUMS. So the IUMS outreach programs include program of educational outreach to developing countries and their microbiologists. Now, for us, although we are on the path of development and will soon be declared developed, as communicated and promised by our honorable Prime Minister of the country, but we still are a developing nation. So as part of the developing nation, we can participate in these IUMS outreach programs. And highlight, which I'm going to put here, is the 6th IUMS outreach program on food safety, which was conducted in Brazil. And as you can see, there are almost 120 participants in this particular program, this outreach initiative that was conducted by the IUMS. Now, the best part is to participate in these IUMS outreach programs. You make an application that you want to host an outreach program in your country, design a curricula, and send it to the IUMS. And if the IUMS executive body decides that the application is worthy of consideration and sponsorship, they will host this outreach program in our country. So we as MBSI target to host or make an application at least to host one outreach program within this year to this IUMS. There are also other initiatives and meetings which are supported by the IUMS. As you can see, the International Yeast International Basic Sciences and Sustainable Development Conference 2022-23 was supported by the IUMS. An upcoming conference on Klebsiella epidemiology and biology symposium in November 2024 is being supported by the IUMS. The IUIS, or the International Congress of Immunology Society, is being held and supported in Vienna by the IUMS. The International Conference on Yeast Genomics and Molecular Biology last year was also supported by the IUMS. And A Life on Our Planet Depends on Microorganism Conference was also supported by the IUMS. And currently, the ASM Microbe, which is supposed to be the largest congregation of microbiology from across the Americas, is being supported by the IUMS and currently being held in Atlanta, Georgia, USA. So these are the benefits of becoming a part of the IUMS. In addition, there are certain awards which are only applicable, or application nominations can be sent by IUMS member societies, and two specific awards like the ARIMA Award for Applied Microbiology, which is given to an individual deemed to have made outstanding contributions to applied microbiology, and the Stuart Mudd Award for Studies in Basic Microbiology, which is also a recognition of outstanding selfless endeavors and contributions in microbiology for the good of society. And I'm sure MBSI as a society will apply for this Stuart Mudd Award at least for this year and see whether our nominations are successfully claimed. There are other member benefits of becoming a member of the MBSI. The first one is you make yourself heard. There are a lot of problems we are facing in India, which are governed by international rules and regulations, because of which we are not able to participate or contribute to the development of microbiology across the globe. So we will have our voice heard by getting a representation in international committees. I will come to that in a couple of minutes. We get voting rights in these bodies. We submit nominations for these awards. We participate in outreach programs. We solicit sponsorships and fundings for our own activities. And we get discounted registration. And lastly, and the foremost and the most important is represent microbiological sciences at the international platform, which is the major objective of MBSI as well. To take you around the structure of IUMS, the structure of IUMS is primarily governed by the General Assembly, which is formed by the members and the associate members and their representatives, which elects an executive body, and the executive board has a committee of the public policy, which basically decides the entire functioning. Now, the IUMS consists of three divisions: the bacteriology and applied microbiology, which is BAM, mycology and eukaryotic microbiology, which is MEM or MEM, and the virology division. Within these three divisions, there are 6 committees, 9 commissions, and 2 federations, together called as COMCOFs, under which are subgroups consisting of working groups concerned with bacteriological, mycological, and virological research. Now, these committees, commissions, and federations are bodies that govern different aspects of microbiology. I'll quickly go through the MEM and virology first, and then move to the bacteriology and applied microbiology later on. So the M division comprises of the International Commission on Food Mycology, International Commission on Penicillium and Aspergillus, the International Commission on Taxonomy of Fungi, and the International Commission on Yeast. The virology division comprises of the ICTV, or the International Committee on Taxonomy of Viruses. Now, we being a pure microbiology society, the IUMS has put us under the BAM. The BAM is the Bacteriology and Applied Microbiology division, under which there are 3 commissions or committees. First one is the International Committee on Food Microbiology and Hygiene. The second one is the International Committee on Systematics of Prokaryotes. The third one is the International Commission of Microbiological Specifications for Food. Now, these 3 commissions regulate different rules for microbiological specifications of food. Committee on Food Microbiology and Hygiene decides the regulations that are involved in production of food products. The International Committee on Systematics of Prokaryotes decides the codes of nomenclature and applies them across the globe. The WFCC, the World Federation for Culture Collections, is a prominent body that actually is a conglomerate of culture collections across the globe. And luckily, there are a couple of culture collections from across India which are part of it. The microbial, the collection of microorganisms, MCM, which I represent, is a part of WFCC since 1981, and it was recently recognized as an affiliate member of WFCC. So we are part of WFCC, WDCM, which is the world database for collections of cultures of microorganisms. The ICB, which is the International Committee on Bionomenclature, decides how societies across the IUMS will be dealing and using the society resources. So these are the 5 commissions and committees or federations which are part of the BAM. Luckily for us, we can get nominated in 2 of these committees. The first one is the ICFMH, which also has a place for an Indian member. We have not yet nominated any Indian member of MBSI as a representative to ICFMH, simply because we have not heard back about the procedure to nominate a member. For the ICSP, the Microbiology Society India has nominated me as a full member of the ICSP, and because of which I was able to participate, I am able to participate in the decisions that govern the systematics of prokaryotes. So I'll move gears and move to the ICSP. The ICSP, or the International Committee on Systematics of Prokaryotes, was originally conceived as the International Committee for Bacteriological Nomenclature in 1930 in Paris. In 1936, mostly in 1936, a proposal was floated to change the name of the committee from International Committee for Bacteriological Nomenclature to the International Committee on Systematic of Bacteriology, which is the ICSB. Now, mostly in 1936, this proposal was adopted and the name was changed. However, looking at the diversity of bacteria, archaea, and new life forms that were considered at that time, the name change was again proposed in 1999 in Sydney to the International Committee on Systematics of Prokaryotes. And the name was adopted in 1999. You can read more about the ICSP at www.the-icsp.org. Now, as you know, prokaryotic nomenclature is governed by the ICSP. The names given to prokaryotes are regulated by the International Code of Nomenclature of Bacteria, which is now called as the International Code of Nomenclature of Prokaryotes. The body that oversees the nomenclature of prokaryotes is called the ICSP, which is formerly known as the ICSP. All right. So these are the 2 distinctions that the Code of Nomenclature of Prokaryotes decides the rules, and the committee that formulates these rules is the ICSP. Now, luckily for us, again, as you can see, members of ICSP from India, Dr. Yogesh Shauche from INSA was the earlier nominated member from INSA, and he's continuing to be there. And myself, we've been nominated through the MBSI to become a part of this ICSP. And to take you back to the structure of the ICSP, the societies and committees behind the ICNP form the most major group, within which is the IUMS, within which is the BAM, within which is the ICSP. And the ICSP has judicial commissions, which actually form the part of the major. We have a disturbance. Can you mute yourself, please? Okay. Sorry for that disturbance from somebody. As I said, the societies and committees behind the ICSP are the main driving force. And we, as one of the members of the of the IUMS, are also part of the ICSP now. And because of which, we can now take part in the functions of the ICSP. The major function of ICSP is to represent the diversity of interests of different microbiological disciplines on matters concerning the nomenclature of prokaryotes, to write, edit, interpret, and disseminate the ICNP, to establish publications deemed necessary for advancement of systematics of prokaryotes, which are primarily the approved list of bacterial names, or the validation list, and the IJSEM journal, in which all new taxa are typically described. To elect members of the Judicial Commission, to consider and vote on all recommendations, to receive reports of the subcommittees on taxonomy, to organize and sponsor regional, national, and international conferences, to elect the executive board, to appoint as life members of the ICSP, and to take other actions that ensure the proper application of the code. Now, all of these revolve around the code. And as the committee's primary responsibility is to formulate, write, edit, interpret, and disseminate the ICNP, I will primarily focus on the ICNP. So originally, the International Bacteriological Code of Nomenclature separated from the Botanical Code of Nomenclature in around 1947. The paper was published in 1948, the first code of bacteriological nomenclature. So this was then has undergone several revisions. 1958 is was called the International Code of Nomenclature of Bacteria and Viruses. 1966 to 1992, it underwent 3 revisions. It is called the International Code of Nomenclature of Bacteria. And in 2008, following the 1999 recommendation from Sydney, the code was changed to reflect the code of the name of the committee, and it was called the International Code of Nomenclature of Prokaryotes. And the most current revision, which was published in 2023, is the 2022 revision of the ICNP, or the Prokaryotic Code. Now, how does Indian microbiology benefit from being a part of the ICSP? Obviously, make yourself heard. We know that there are several problems that are being faced by microbiologists in India. So we will raise these concerns into the ICSP, which are primarily the body that governs the rules, and request them to reconsider some of the changes that we propose so that it becomes easier for us and for the world as well. So you make yourself heard, you get voting rights in different decisions that are made by the ICSP. The inability to publish in the IJSEM due to implications of Rule 30, that is one of the major responsibilities that I have taken, along with several others from other countries who are facing a similar problem. Coordinate between national and international bodies to revise existing regulations. Submit nominations for various awards of the ICSP. And finally, represent microbiology at the world stage. So this is one of the primary. And Muskan, could you please mute yourself? So this is something that is of primary objective for us to be a part of the ICSP. So I'm going to focus a little on the inability to publish in the IJSEM due to implications of the Rule 30. Now, there are several types in a nomenclature code, and a type could be a fossilized species, it could be a preserved specimen, it could be a live culture, or it could be a specimen in, in documents. Now, in 1966, as you can see, the type of a species or subspecies is preferably a designated type strain, or in special cases, it may be a description, a preserved specimen, or a preparation, or an illustration. In 2008, this was revised to a viable culture, which has to be deposited in at least 2 publicly accessible culture collections in different countries. Now, this proposal, although the code was published in 2019, it was effective from 2008 because it was revised at that time. And since 2008, because of certain regulations, Indian microbiologists are not able to deposit cultures in 2 publicly accessible culture collections. And the reason for that is that the National Biological Authority, which is the Biodiversity Board, does not, I should use a correct word, it doesn't prohibit, but it puts additional regulations and checks for cultures to be deposited in foreign culture collections. Now, one of the primary conditions to fulfill Rule 30 is that the culture has to be freely accessible by anybody who wants to use it for research. However, the NBA requires that whenever any foreign culture collection has a request for supply of that culture by anybody, the foreign culture collection must notify and seek permission. Sorry, not the foreign culture collection, but the person who is requesting the culture from a foreign culture collection must approach the NBA India and request prior approval. And this condition is considered restrictive by the ICNP or the editors of IJSEM. And for that reason, they deny that the certificate of deposit issued by that particular culture collection does not fulfill the requirements of Rule 30. And because of which, we are not able to publish in the IJSEM. Now, mind it, IJSEM is a body of editors or the journal which is primarily published by the ICSP. So there are certain other problems with handling prokaryotes. Most prokaryotes have not been brought into pure culture. So most cannot be validly named under the ICNP. A recent proposal has been floated where you can name such organisms which cannot be cultivated as candidatus. The second one is that many organisms are fastidious and cannot be maintained in culture collections, especially like fastidious organisms, anaerobes, extremophiles, alkaliphiles, barophiles, certain hard to cultivate organisms like acidobacteria, verrucomicrobia. So all of these organisms cannot be maintained by every culture collection. Only few culture collections in the world have this capacity to, or the expertise to preserve these organisms. One of the examples that I highlight here is Nitrosopomilis maritimus. This was isolated in 2005. The genome could only be sequenced 5 years later, and it could only be validly published in 2017. So that means it was almost a 12-year gap to publish this particular organism under the ICNP. Then there is this third problem, as I said already, about the regulations to fulfill the Nagoya Protocol, to take care of local legislations, and to take care of the rules and regulations of the ICNP, under which our cultures, when they are deposited in foreign culture collections, they are not accepted as free use. So because of which we are not able to publish. Luckily, as soon as we became a member of the ICNP, there was a meeting that was just conducted 2 days ago on June 13th, where I participated. There were around 26 members from the ICSP membership that participated in this meeting. And we had a huge debate about this particular topic. There were several supports that we received, especially from Brazil, South Africa, and other nations that are facing similar problems. So this meeting lasted roughly about 2 hours, started at around 8:30 day before and finished by around 10:30 in the night. There were a lot of discussions, and eventually people have considered our concerns. We've raised them, and hopefully in the next several meetings, we will see how things progress. We would like to gather more support to resolve this. Now, there are few certain changes that have happened in the ICNP. The first and foremost I would like to highlight that happened in 2021 is the valid publication of the names of 42 phyla of prokaryotes. The proposal to include the rank of phylum was floated in 2015, and in 2018, a suffix OTA was recommended, and in 2021, this valid publication of the names came in effect. And what it has done, it has changed how you name phyla. So actinobacteria are now named as Actinomycetota. Firmicutes are now named as Bacillota. Proteobacteria are now known as Pseudomonadota. Now, these changes are not so severe, but people who are working in microbial ecology have had a lot of friction in the initial days. Luckily, people are accommodating to these changes, and these changes are now being adopted throughout the world. So there are certain names which are validly published under the ICNP. This is maintained by the list of prokaryotic names with standing in nomenclature, or the LPSN website. There are a total of 24,876 gene species which are now listed under the ICNP, 4,146 representing these many genera, roughly 780 families belonging to 53 phyla, and which are then divided into tribes, suborder, subclass, and genus. So these are the names of the candidates which are already validly published under the ICNP. So we have roughly around 2,600 species designations. We have close to 1,100 genera, genus designations, around 246 families, representing 201 phyla. So as you can see, there are 53 phyla which are cultured in the lab, and there are 201 phyla which are not yet cultured in the lab, or at least not yet, presented as without candidatus status. So that clearly indicates that there is a vast majority of the bacteria, archaea domain which are still yet to be cultured in the lab. And to circumvent these problems, a parallel code called the SEQ code was proposed in 2022. And the SEQ code is a nomenclatural code for prokaryotes described from the sequence data. Now, there is a lot of debate happening in the systematics and taxonomy community regarding the application, feasibility, futuristic perspective, and how the two codes will survive in parallel. Which one is formal, which one is informal. There are a lot of debates happening. And we, as part of, as I said, I'm also part of the Burgess International Society for Microbial Systematics. And that is another organization that is working towards, resolving this conflict and working towards the future of microbial systematics. It's an advocate, international advocate, and BISMIS basically conducts a live session every month on the third Saturday. So in the last month, in the month of May, we hosted Professor William Whitman, Louis Rodriguez, and Maria Chuvacina, who basically countered their concerns against the new proposal and amendments for further integration of the candidatus into the code. And they suggested that it is not the best of the both worlds for the cultured as well as the uncultured community. Now, you can scan this QR code to visit the YouTube seminar that is recorded live and present there. To have a better representation of what the code proposes, a new session which is coming up on coming Saturday, June 22nd, is also proposed by the members of the ICSP, who are the authors of that paper, Professor Ed Moore, Professor Dr. Marcus Coker, and Professor Henrik Christensen, who will be participating in this discussion. And they will be presenting the side from the ICSP as to why this is the best of the both worlds. Now, if you would like to read more, you can visit the BISMIS website, or you can visit this registration page, scanning this QR code, and you will be able to identify where all these things are taking us. So now that I'm talking about BISMIS Live, I would like to just quickly ask you a question. I want to understand how many of you attend BISMIS Live or have seen the YouTube recordings? If you can, just quickly unmute yourself and say yes, I would understand that.
I attended, sir.
Oh, great. So at least there are a few people who actually attend the BISMIS Live. Now, there were many, when I asked this question, very many who actually did not know that a society like the BISMIS exists. And this was also a part when we proposed the membership of MBSI to the IUMS. IUMS was under the impression that there is a single society from India which is already part of the IUMS. They did not know that the Association of Microbiologists of India existed. They did not know that the Microbiology Society India existed. So there are cases where such societies exist, and they are working for the promotion of the field of microbial systematics. So a brief, about BISMIS. It was conceived in 2006, and finally in 2011, it was constituted as BISMIS. The goal is to promote research in microbial systematics. It acts as a global communication and advocate for research efforts on microbial systematics. Now, my personal association with BISMIS started back in 2007. This was a formal association in 2007. My actual association with BISMIS started in the year 1997, when I actually joined the first year bachelor's course in microbiology in Nashik. So my BSc, when I first learned about what Burgies is, I was fascinated with this field. So I was a volunteer for the trust meeting, for the Burgies trust meeting in 2007, then became a charter member for the establishment of this society in 2010, became the secretary of the body in 2014 and continued until 2018, when I became the vice president of the society, or the president-elect in 2018, continued until 2020 as the president-elect, and took up the presidentship in 2020 and continued until November 2023, when I handed over the presidentship to my colleague, Professor Wenjun Li from China. And today I'm still an integral part of BISMIS. I'm an integral part of the MBSI, and because of this cross association, we are able to harvest the benefits of being recognized by several people across the globe. So we have a lot of BISMIS Live speakers. This is the first year speaker, second year speaker, third year speaker, and we are currently in the fourth year of BISMIS Live. We have a lot of sessions. The next 3 sessions are hands-on workshops, which are free for everybody to attend. This is primarily focused on sequence analysis, metagenomics, and microbial genomics. So 3 sessions completely dedicated to free hands-on workshop for masters, PhD students, and undergraduates. And this is the beauty of our membership. The membership of BISMIS is roughly about 42% of master student. And mind it, most of these are from India. And most of these became members or are participants of BISMIS because of MBSI. Because my dissemination of the sessions is primarily through MBSI, and all of these students are participating. And there are few success stories of BISMIS Live coursework. The sessions of BISMIS Live are now being used as coursework material across the globe. Shivaji University in India, in Sangli, is already using some of this material as coursework. I personally use some of these lectures as assignments for the University of Pune. And Brian Hedlund from the University of Nevada, Las Vegas, is also using the material as coursework material. So this is one of the images that I took in Sangli, where we conducted a workshop on the C code. And through the C code and through MBSI, I want to share, that MBSI is one of the most approachable, outreach intensive, and open to new ideas kind of a society, because of which we are now able to reach the heights of representing ourselves on the international platform. And with this, I would like to close and promote my lab website, Microbial Lab. I'm looking for PhD students in my lab. So if you have a JRF, if you have any other fellowship, exam qualified, connect with me, and I'll be happy to take you as a PhD student in my lab. Thank you so much. And I'll be happy to take questions.
Thank you, sir. Thank you, Dr. Kamlesh, sir. And thank you, Dr. A.M. Deshmukh, sir, to introduce us IUMS and what is the IUMS and ICSP. So please, participants, I request to you, if you anyone have queries, please discuss with sir.
There are two questions in the chat box. In the chat box, Kamlesh sir, please
Yes,
go
sir.
through the chat box.
Let me see, sir.
One by Prakash Halami.
Yes, I'll take these questions one by one. Does NBA allow to deposit bacterial cultures in overseas culture collection centers? The question has been asked by Professor Prakash Halami from CFTRI, and the answer to this is yes. There is a Form C that needs to be submitted to NBA.
Okay.
And the submission of Form C, once the NBA is notified, you can then submit your culture to the overseas culture collection along with the Form C copy that you have notified the NBA. And it depends on the culture collection whether they will consider it restriction-free or restricted access. So currently there is only KCTC, Korean Culture Collection, and CCUG that accepts cultures. The only thing is they accept culture, but they consider it to be restricted. So their certificate is kind of nullified by the editorial board of the IGSEM. So because of which we are not able to publish in IGSEM, we have to publish in some other journal and then get it effectively published later on in the IGSEM. Does that answer your question?
Yeah, to some extent, quite convincing only. Otherwise, also ATCC we used to submit a long back, but now presently since NBI restrict in several fronts, so whether there is any new guidelines. And one of the foreign company actually approached us to take a probiotic culture. So major hindrance was again from the NBA because they wanted to use for commercially.
Yeah, so NBA has a protocol to be followed. It is just a bit lengthy. So the person who is requesting a culture has to approach the NBA with the specific forms and the application fees which are listed on their website. Yeah.
And
so if those approvals are given and the benefit sharing is, equitable, then NBA doesn't have any problems.
Yeah, just on the similar line, if any Indian company also wants to purchase a culture, so NBA comes in a picture for any approval?
Sorry, there was some disturbance. Can you repeat?
If any other company wants to explore our culture for a commercial purpose, does NBA comes in a picture for permission?
Absolutely.
Mm-hmm.
As a culture collection, you can supply cultures to Indians within India without any restriction for research. The moment the commercial aspect comes in, you have to notify NBA, and NBA will then decide what is the equitable benefit and access and benefit sharing.
So notification has to go from the researcher or from the company?
From the person requesting the culture.
Okay, okay. This is quite clear.
Yeah.
I think there is one more question from my side, I guess, on the chat box.
I'll go ahead, sir.
Yeah.
I'll take those. The next question is from Prasoon Kumar. How about 2 culture collections in India? Can we submit the culture there? Yes, you can freely submit culture collections to cultures to any number of culture collections. The only problem is you have to have at least one foreign culture collection where your culture is available. This is to ensure that in case of any mishap in a culture collection, the culture is freely available from a different culture collection, and those at least 2 culture collections have to be in different countries. So one can be in your country, one can be a country outside, and that is where the whole problem is. We'll move to the next question. The next question is again from Professor Prakash Alami. How does non-culturable and newly identified genera are named? Okay, so Dr. Prakash, there is the same debate that in the very recent proposal that has come in the IJSEM, the further integration of the candidates into the ICNP. What it proposes is if you're not able to grow an organism, you can do the genome sequence and then you can submit to IJSEM, but with a candidatus designation. And you can get it pro-validly published. This is still in proposal stage. A debate is currently going on. And on 5th of July, the voting on this proposal will open. The voting will be there for 30 days, after which a final decision will be made. So I would strongly suggest that you watch the previous session that was held on the C code and the new session that is upcoming on June 22nd by the ICNP. So since the time the C code has come, C code was originally proposed to be a part of the ICNP in 2015 when a proposal was made to recognize DNA sequences as type material, so that all of this problem would have been taken care of. But then at that time, the ICSP membership voted against this proposal, and this proposal was rejected in 2019 or 20, I believe. And finally, in 2020, 22, the C code was proposed. And since the time the C code has been proposed, there are 2 schools of thought. One that believes that C code is more open. The other that believes that having 2 parallel codes will create confusion into the nomenclature. And there are, negotiations going on or different modifications going on. And the proposal by the ICNP to further integrate candidatus is one of the solutions proposed to take care of the problem of organisms and Indian cultures which cannot be submitted to ICNP as valid publication. So with this new proposal, one can submit, but they will still be called as candidatus. They will not be called as genus and species. They will be called as candidatus, genus, and species. So the name candidatus will always be prefixed before the genus and species epithet. So that will always be there. In future, when your culture is isolated, identified in the lab by somebody else, and that particular author is able to submit it in 2 culture collections, at that time, the person will be responsible to carry forward the pre-existing name and the pre-existing authorship of the original authors who named that. So that is a kind of a positive move towards it. So all of this is now geared towards how this voting will happen and what will be the outcome of this vote that happens in the month of June. And we as MBSI will have one voting right because I am a member now. So we will raise the voice, collective voice of whatever we decide. Okay.
But that culture traceability is only at a DNA sequence level, right?
No, once the culture is isolated, the type material will change. The
Okay.
type material will not be the sequence anymore. The type material will become the culture.
Yeah, but you don't have a culture cultivation media. How you would do that?
Today you may not have, but in future, if that culture is isolated,
Okay.
identified, and grown in the lab, that time the genome sequence will be replaced by the new type material, which is the culturable organism.
Wonderful, wonderful. Thank you.
And again, one more question from Dr. Prakash is what type of R&D in your lab goes? As you could grasp from my presentation, my main focus is microbial ecology, systematics, and taxonomy. So my web page, microbiallab.org, will describe all the work that goes on in my lab. I mostly work with genomics, metagenomics, and transcriptomics. And currently, the lab is geared towards bioprospecting the cultures that are available within the culture collection, and also taking care of these other aspects of my research. So we also work on quorum sensing. We also work on microbial community dynamics. We try to develop theories from real data and see how progression of microbial communities take place in the natural ecosystem, and how these models can then be implemented to restore denatured or degraded ecosystems. So that is the approach that we are using.
Wonderful. Thank you.
Vidya Sonalkar is asking, will you please elaborate on Nagoya Protocol? So Nagoya Protocol is basically a set of recommendations, and India is a signatory to this protocol, which basically decide the access and benefit sharing of biological resources. Now, that biological resource could be a pure culture, could be a plasmid from an organism, it could be anything of biological nature. Now, this means that the place from where it has been isolated, the person who has isolated, the country to which it belongs to, whenever something is commercialized out of it, all parties that are part of the development or bringing of that culture or biological resource into application must obtain benefit from that commercialization. So that is the basic crux of the Nagoya Protocol. I hope I'm clear on that. A question has come. Yeah.
Thank you, sir.
Yeah. A question has come up. How will Indian culture collections and IDAs benefit from this membership? Yes. The Indian culture collections and IDAs will benefit from this membership because now at least we have a voice. Earlier, there was a single voice. Now we have two voices. And because of two collective voices, obviously, the impact and the maneuverability or taking people in confidence will be much more easier. We are trying to resolve this problem of how Indian culture collections or cultures that are available in India can be recognized by the culture collections abroad. There are efforts that are ongoing at different dimensions, different directions, and these efforts will definitely benefit the culture collections in general. Vidya is asking, will you allow to work in your lab to the person registered in other university? It really depends. If the person is coming for a short term on a collaborative basis, it is allowed, provided it is official. Otherwise, there are master students who come for summer training, who come for dissertation projects in my lab. They can definitely come. somebody with Snehal's iPhone is asking if we want to get metagenomics facility for sample analysis, will your institute help and at what cost? So the costs are listed on our culture collection website, and definitely help will be provided in doing the sequencing, in doing the bioinformatic analysis, and everything is built into the entire service package. So that is always there. As a culture collection, we are involved in all the activities that are typically provided by a culture collection: identification, characterization, detailed analysis, description of novel species, consultation. We being a specialized culture collection in anaerobes and extremophiles, we also conduct an annual workshop of 7 days, which is only dedicated to hands-on training in anaerobic microbiology. So we conduct this annually. We also conduct a workshop on identification, characterization, and preservation of microorganisms. We also conduct an online workshop on DNA sequence analysis for microbial identification. So we have these three workshops that are typically conducted throughout the year. So you can connect and visit our website at mcm.aripune.org. So that is the website where you can visit. If there are no more questions, I'll hand over the proceedings to Dr. Deshmukh and
Thank you.
coordinator Aparna.
Thank you very much, sir.
My pleasure, sir.
Yes, because you see, I am the president of MBSI, but I was not knowing. Only through you, I believed on you, and
Thank
I
you so
have given
much.
a green signal to you.
Yes,
But today, I came to know that what a great achievement we had by entering into IUMS.
right, sir.
And we'll do many things in the future. I will understand slowly and slowly functioning of IUMS and will enter in many activities of IUMS.
Right.
So, okay, now I request Aparna for vote of thanks. Aparna.
Thank you, sir. Thank you so much for your insightful and engaging presentation. Your expertise and passion truly made a significant impact on us. We appreciate the time and effort you dedicate to sharing your knowledge. Thank you so much, sir. Our next session will continue with new topics.
Thank you. Thank you, sir.
Thank you. Thank you so
Thank
much.
Yes, and now this lecture is recorded, will be circulated for the interested person.
Thank you.
Thank you.
Thank you, everyone.